I want you to just imagine a single, perfectly round, completely featureless ball of cells.
Okay.
You're looking at it under a microscope, and it looks totally uniform. I mean, every cell appears virtually identical to its neighbor.
All right, just a blank slate.
Exactly.
Yeah.
But somehow within this tiny microscopic sphere, this staggering geographic coordinate system is, already firing up.
Yeah.
Like, how does one cell sitting on the left side just know with absolute molecular certainty that it has to become part of a beating heart ventricle?
Yeah, and how does the one right next to it know it's going to be an eye?
Right. Just a few micrometers to the right.
It is, it's the ultimate architectural mystery in biology. We are talking about the exact moment a generic cluster of cells breaks its symmetry and decides to actually build a complex three-dimensional body.
And today, our deep dive is taking us straight to the frontier of that exact mystery. So we are unpacking this massive 2023 paper from Nature Genetics.
It's a huge one.
It really is.
Yeah.
The researchers here have essentially built like a Google Earth for the developing embryo. We're looking at spatiotemporal transcriptomic maps of mouse embryos right at the onset of organogenesis. So specifically embryonic days 8.5 to 9.5.
Yeah, and to really appreciate the sheer scale of what this team pulled off, we have to talk about the massive blind spot that developmental biologists have been dealing with for years.
Oh, totally. The blender problem.
Right, the blender problem. So if we wanted to know what genetic instructions were active in a developing embryo, we relied on single-cell RNA sequencing, which, gives you incredible detail about individual cells.
There's at a huge cost.
Exactly. To get that detail, researchers had to literally dissolve the embryo. They used enzymes to break it down into this microscopic cell soup just to sequence the RNA.
Right. So it's like having this beautiful 1,000-piece jigsaw puzzle of a landscape. You want to understand what the puzzle is made of, so you dump all the pieces into a blender, hit pulse, and then pull out individual pieces to analyze them.
Yeah, which sounds crazy when you put it like that.
It does, because you can look at a piece and say, okay, this piece has blue on it, so it must be the sky. But you completely destroy the geography. You have absolutely no idea where that piece of sky belongs in relation to, like, the mountain or the river.
You lose all the spatial context. And in embryonic development, context is literally everything. A cell's physical location dictates its fate.
Right. It's talking to its neighbors.
Exactly. It receives chemical signals from its neighbors based entirely on where it's sitting. So the big breakthrough in this paper is a piece of technology called Slide-seq.
Which is where we finally get to take a high-resolution photo of the puzzle while it's still fully assembled.
Yes.
So how exactly are they pulling this off without blending the embryo? What's the actual mechanism?
The methodology is brilliant. They took these embryonic day 8.5 to 9.5 mouse embryos and sectioned them into incredibly thin sagittal slices. I'm talking just 10 micrometers thick.
Wow, so thin.
Very thin. And they placed these slices onto specially engineered surfaces that are packed with these microscopic beads. Now, here is the genius part.
Hey.
Every single bead has a unique DNA barcode, and the researchers know the exact physical X and Y coordinate of every single barcode on that surface.
Oh, I see. So when the tissue slice sits on these beads, the RNA from the cells just bleeds out and gets tagged by the bead directly underneath it.
Exactly. They captured data from over 533,000 of these high-quality beads. Each bead grabs the messenger RNA, the active genetic instructions, from the cells it physically touches.
Because of that barcode?
Right. Because of the barcode, they can map every single piece of RNA back to a precise physical coordinate. They are mapping the entire active transcriptome without losing the geography.
But they didn't stop at just two-dimensional slices, right? Because they developed this custom computational tool called C3D.
Yeah, taking 2D slices is great, but obviously embryos are three-dimensional. So the C3D software takes all those ultra-thin Slide-seq slices and uses these spatial registration algorithms to just stack them back together.
Like a loaf of bread.
Exactly like a loaf of bread. It aligns the data perfectly to reconstruct interactive, fully three-dimensional virtual embryos.
They literally brought the embryo into the digital world. And with this digital embryo, the paper highlights this technique called VISH, virtual in situ hybridization. Let's explain why that's such a massive upgrade. Because traditional in situ hybridization is, it's grueling.
Oh, it is. It's so tedious.
You take a physical sample, use a chemical probe to stain it for one, maybe two genes, and see where it lights up under a microscope. It takes days.
Days. And you burn through your precious tissue samples very, very quickly. With VISH, because the entire transcriptome of over 27,000 genes is already mapped in 3D, researchers don't need probes anymore or physical tissue.
They just type it in.
Right. They simply query their software. They type into a database, show me the exact expression gradient of this specific gene along any 3D axis, and the digital embryo instantly lights up. It is literally a biological search engine.
Okay, so let's do a quick recap for you, the listener, before we move on to what they actually found with this search engine. So bullet point 1, we moved from dissolving embryos into a genetic blender soup to keeping them intact.
Right, using Slide-seq.
Bullet point 2, Slide-seq uses hundreds of thousands of barcoded beads to map exact X and Y coordinates for RNA. And bullet point 3, the C3D software stacks those slices to make a 3D searchable virtual embryo.
A huge leap forward.
Huge. So a quick question to you listening. Imagine having a search engine for every gene in a developing body right on your laptop. What specific organ's blueprint would you look up first? I mean, where would you even start?
Well, for these researchers, the logical starting point was the most critical infrastructure. So they zoomed right in on the earliest stages of the developing heart and the brain.
Let's look at the heart first. When they queried the digital embryo, they saw certain genes acting like glowing spatial beacons in the primitive heart tube. They tracked genes like NICIPA and CRIC.
And when they mapped these genes, the expression wasn't just some random scatterplot. They delineated highly specific, complex developmental axes.
Like drawing a map.
Exactly. We are talking about mapping the anterior-posterior, right-left, and dorsoventral axes. They could actually see the molecular outlines of where the primitive ventricles, the atria, and the outflow tract were going to physically form.
So they're establishing the north, south, east, and west of the heart before it even looks like a heart. And I was really fascinated by how they used the gene CRIC. The spatial map showed that SCRIC expression physically separates the mature, differentiated heart muscle cells from the undifferentiated ones.
It literally draws a geographic line, yeah. A border between the cells that have fully committed to being beating heart muscle and the progenitor cells that are, still waiting for their final instructions.
You can physically see the wave of differentiation moving through the tissue.
You really can.
Okay, but the brain mapping between day 8.5 and 9.5 is where this gets even crazier to me. The spatial data shows the anterior portion of the neural tube splitting into the forebrain, the midbrain, and the hindbrain. But wait, I'm stuck on something here.
What's that?
The paper points out that specific genes are restricted to exact spatial zones before any anatomical structures exist. Like, how does a gene draw a border if the physical walls of the brain haven't even been built yet?
Ah, yeah. So that's called pre-patterning. The spatial restriction of gene expression precedes the physical anatomy. The molecular boundaries are laid down in what looks like just a flat, totally uniform sheet of cells.
It's like zoning laws in a city.
Yes, great analogy.
Like the city council zones an empty dirt lot for commercial use long before anyone actually pours the concrete for a grocery store. The cells sitting in that dirt lot are already restricted by the zoning law.
And the researchers found a stunning example of these molecular zoning laws at the R4 boundary.
Which is what?
That's the exact border between the midbrain and the hindbrain. They looked at two specific genes, AuTICUS2 and GBX2. Now, AuTICUS2 is expressed strictly in the midbrain zone, and GBX2 is expressed strictly in the hindbrain zone.
So they don't overlap.
They are completely mutually exclusive, and right at the microscopic fault line where those two zones meet, a totally different gene, FGF8, is expressed.
So FGF8 is acting like a molecular border guard, just standing right on the line.
Exactly. Now, to understand what is actually happening to the cells living on that border, the researchers utilized a computational method called spatial RNA velocity.
Okay, I really want to dig into this mechanism because the paper leans heavily on RNA velocity. And honestly, it sounds like science fiction. Like it's predicting the future state of a cell. How do they calculate velocity from a static frozen tissue sample?
Yeah, it sounds impossible, but it comes down to the life cycle of RNA. So when a gene is first turned on, the cell produces what we call unspliced RNA.
Unspliced.
Right. This is the raw, unedited genetic transcript. Over time, the cell processes it, it cuts out all the junk sequences, and it becomes spliced mature RNA. And that's what's used to make proteins.
Oh, I see. So unspliced RNA is like the raw material sitting on the factory loading dock, and spliced RNA is the finished product rolling off the assembly line.
That is exactly right. And because the sequencing technology can distinguish between that raw unspliced RNA and the mature spliced RNA, the software can actually calculate a ratio.
Ah,
Yeah. If a cell has a massive amount of raw unspliced RNA for a specific gene, it means that gene is actively ramping up. The cell is accelerating in that direction.
he's building momentum.
Right. But if it mostly has mature spliced RNA, the gene expression is stabilizing or kind of winding down. By calculating this across thousands of genes at once, the algorithm generates a directional vector, an arrow pointing toward the cell's future identity.
That is brilliant. They're essentially calculating the biological derivative of the cell's current state. So when they applied this RNA velocity tool to that ODS2 and GBS2 border in the brain,
Yeah.
what did those vectors actually look like?
They saw dramatically diverging trajectories. So imagine two cells sitting right next to each other on either side of that FGF8 border guard.
Okay.
Physically, they are neighbors in a tightly packed space. But computationally, their RNA velocity vectors are pointing in completely opposite directions. They are accelerating down entirely different developmental highways.
Toward completely different structural destinies.
Oh, yes.
Okay, so let's pause and synthesize this with our second mini recap. Bullet point 1. In the heart, genes act as beacons to draw physical ways of differentiation.
Right.
Bullet point 2. In the brain, genes like ADX2 and GBX2 lay down strict molecular zoning laws before the brain physically folds. And bullet point 3. RNA velocity lets us predict a cell's future by comparing raw versus mature RNA.
Perfectly summarized.
Thanks. So here is a question for you, the listener. Does it change your perspective on biology to know that an organism's fate is locked into a strict geographic coordinate system on a molecular level before anything physical is even built? Like, does that blow your mind as much as it does mine?
It really is profound. And, it leads us to a massive mechanical problem that the embryo has to solve.
Which is?
Well, we've seen how these zoning laws work in the tight, localized spaces of the head and the heart. But how does this system scale when the embryo needs to physically stretch out over a massive distance to build a spine and a trunk?
Right, because that's a lot of physical ground to cover.
Mm-hmm.
And the paper introduces a very specific population of cells to answer this, right? Neuromesodermal progenitors, or NMPs.
Yes, NMPs, these are stem cells sitting at the very tail end of the developing embryo. They are bipotent, meaning they basically have to make a choice between two distinct career paths.
They can't do both.
No. They can either become part of the neural tube, which turns into the spinal cord, or they can become paraxial mesoderm, which eventually turns into the somites, the blocks of tissue that will form our vertebrae and back muscles.
So they are the ultimate building blocks for the body's main axis. But when the researchers tracked these NMPs using the C3D map, they uncovered this massive paradox in how these cells behave compared to how far they actually travel.
Yeah, it was super surprising because normally you would assume that if a cell makes a massive change to its genetic identity, it's probably moving to a totally new neighborhood.
Right. Big genetic shift equals a big physical relocation.
Exactly. But the spatial transcriptome showed the exact opposite. They compared the transcriptional distances, like how drastically the RNA profile changes, against the physical micrometer distances between the cells in the embryo.
And what did they find?
They found that changes in a cell's transcriptome are highly disconnected from its physical travel.
Wait, really? Explain that.
So the NMP stem cells are packed into this tiny, tightly restricted area at the tail end. While they are crammed into this small space, their gene expression changes violently. They lock in their completely different fates, spinal cord or vertebrae, all while physically touching each other.
Oh.
And then, once their fates are totally sealed, they physically stretch out and disperse across the entire length of the embryo's trunk. By the time they are migrating and actually building the spine, their transcriptional differences have kind of quieted down.
That's wild. It's like they endure an intense molecular identity crisis in a crowded room, figure out exactly who they're going to be, and only then do they leave the room to go build the rest of the body.
That's exactly what happened.
Now, as they dispersed to build that spinal cord, the researchers unleashed another new computational tool. It's called axis profiling. Because they have this 3D search engine, they drew a line straight down the neural tube and commanded the software to identify any genes that were spatially co-expressed along that specific physical line.
Right, so they profiled the anterior-posterior axis, which is the length of the spine. That's roughly 4,600 micrometers long in this developmental window. And what emerged was an exquisite sequential patterning of Hox genes.
Now, the Hox genes are the classic fundamental architects of the body plan, right?
Yeah.
They tell the body where to put the ribs, where to put the limbs.
Yes. And the C3D map revealed 6 distinct, highly localized modules of these Hox genes. They trigger in a perfect collinear sequence.
Like dominoes.
Exactly. As you move from the neck down toward the tail, you progress strictly through Module 1, then Module 2, all the way down to Module 6. It is a flawless barcode of development, laid out in physical space.
But the length of the spine was just the beginning. They also profile the dorsiventral axis. So this is the axis running from the back of the neural tube down to the belly side. And this axis is incredibly narrow, like only about 320 micrometers across.
Yeah, tiny space.
Yeah. But when they mapped the known genetic markers here, everything was exactly where the textbooks say it should be. Genes like Shoshonic hedgehog were perfectly localized down in the ventral floor plate.
Which is great because finding the known markers is crucial. It validates the accuracy of the Slide-seq technology. But, the real power of having a completely unbiased whole transcriptome map is finding what you weren't looking for.
Yes. This is my favorite part. Because of the extreme resolution of this tech, alongside those known markers, they discovered 43 brand new, previously unannotated genes.
43?
Yeah, 43 new genes that show highly distinct patterned expression along that tiny 320 micrometer axis.
This is a major revelation for developmental biology. I mean, we thought we had a pretty solid understanding of how the floor plate and roof plate in the neural tube communicated. But finding 43 new localized actors means there are massive pieces of the signaling puzzle that we have been completely blind to until now.
Right, and think about the clinical implications of that. If we now know the exact spatial coordinates of 43 new genes involved in wiring the embryonic spinal cord, that is 43 new potential targets for understanding and treating severe neural tube defects.
Absolutely.
Okay, let's hit our third mini recap. Bullet point 1. Stem cells, called NMPs, decide their fate in a crowded space before spreading out to build the spine.
Yes, the identity crisis.
Exactly. Bullet point 2. Axis profiling revealed perfect sequential barcodes of Hox genes along the spine. And bullet point 3. Researchers discovered 43 brand new genes patterned along the dorsiventral axis.
Spot on.
So, listener, consider those 43 newly mapped genes. How might understanding their exact spatial coordinates unlock new treatments for spinal cord defects? Just something to think about as we move into our final section, because investigating those structural defects brings us to, honestly, the most dramatic finding in the whole paper.
Yeah, it really is, because we have spent this whole time marveling at the perfect, intricate choreography of a healthy, wild-type embryo. But what does this high-resolution geographic map look like when the fundamental rules of development just completely break down?
Right. To find out, the researchers ran their C3D pipeline on a mutant, specifically a TBI6 knockout mouse. Let's explain what TBI6 actually does.
So TBI6 is a gene that is absolutely critical for mesoderm specification. Remember those NMP stem cells we talked about earlier?
The ones that have to choose between becoming neural tube or becoming mesoderm.
Yes. TBI6 is the master switch that forces them down the mesoderm pathway. If you knock out the TBI6 gene, that developmental choice just completely breaks down.
And the anatomical result of this knockout is incredibly bizarre. Like, a normal embryo has one single neural tube running down its back. But in the TBI6 mutant, the embryo grows 3 neural tubes.
3 of them?
Yeah. It has a central tube and then 2 extra ectopic tubes growing parallel on either side.
Morphologically, it's a catastrophic defect. But in the past, without spatial transcriptomics, researchers could really only look at the physical shape of these 3 tubes and just, guess at what was happening inside them.
Right. They had no map.
Exactly. But with Slide-seq, this team could finally see the molecular geography of the mutant tissue.
So what did the digital map actually reveal about the cells trapped inside those extra tubes?
It revealed a profound cellular identity crisis at the transcriptional level. The cells inside the 2 extra tubes are expressing heavy neural markers, like SOX2.
Which makes sense, given they physically look like neural tubes.
Right. But simultaneously, deeply embedded within the same spatial coordinates, these exact same cells are heavily expressing mesodermal genes, like ALDA1A2.
So they are trying to be 2 completely different tissues at the exact same time.
Yes.
It's like, okay, imagine a massive construction crew has been hired to build a skyscraper. That's the neural tube. But someone in corporate messed up the distribution, and half the crew on the site was handed blueprints for a steel suspension bridge. The mesoderm.
Oh, that's a great way to put it.
Right, so you have electricians trying to wire elevator shafts on the exact same floor where guys are trying to anchor massive steel bridge cables. They're both building furiously, right on top of each other, following completely contradictory instructions.
That is exactly what the transcription data shows. And furthermore, when the researchers looked at the dorsoventral patterning in those extra tubes, the axis where we just talked about finding those 43 new genes,
Right.
the map showed that the patterning was only partially complete. The molecular gradient was broken. It's just this bizarre, unstable hybrid structure. And because of this paper, we aren't just observing the strange anatomy of a mutant anymore. We have a high-resolution map of the exact genetic glitch that caused it, plotted across three-dimensional space.
It really is a triumph of methodology. I mean, we started with the old paradigm of breaking the embryo down into a blended cell soup just to guess what genes were there.
Yeah.
And now we have a fully searchable high-resolution 3D hologram that maps the precise geography of life forming in real time.
The ultimate synthesis of this paper is that it provides an entirely new framework for developmental biology. Slide-seq, combined with the C3D algorithms, proves unequivocally that a cell's fate isn't just determined by the genes it contains.
It's dictated by the exact spatial coordinates of where that cell is located.
Exactly. Where it's located and who it is talking to. And for the first time, we can actually observe that conversation in its full, unaltered context.
It fundamentally upgrades our understanding of the building blocks of life. So as we wrap up this deep dive into the architecture of the embryo, I want to leave you, the listener, with one final provocative thought to mull over.
Lay it on us.
If we have now reached the point where we can map the exact geographical three-dimensional coordinates of every single gene activating during the onset of organogenesis, how far away are we from being able to reverse engineer this map?
Oh, wow.
Right? If we know exactly where and when the molecular zoning laws are laid down, could we one day feed these exact spatial coordinates into a biological 3D printer loaded with human stem cells? Could this digital Google Earth of the embryo be the actual blueprint we need to perfectly grow complex, functional human replacement organs right in the lab?